This is a production of Cornell University.
So I started at Cornell fresh out of grad school. This is July 1988, probably before a lot of you were on the face of the earth. And when I started, the cooperative extension specialist here told me that growers were having trouble controlling powdery mildew. They weren't getting as good control as they had in the past. Pumpkins were a new and up and coming crop. This was the beginning of the U-pick and the agri-entertainment time. So an important crop to be working on. One of the big advantages that I've seen in working here on Long Island is the close interaction with cooperative extension. So it's been really a huge asset to be for me to be on top of what the problems are for growers, to work with extension on management, on research together, getting information out to growers. So it's a really great a place to be located. You can see the characteristic powdery mildew symptoms on the leaves around me in this picture. That white growth is predominantly wind dispersed spores. So if you use targeted fungicides applied weekly, top of the slide, you'll see really great control. A lot of good green leaves here in early September, and here's non-treated control. And your first thought might be, well, powdery mildew is only affecting the leaves. Why is it important to control it in a crop like pumpkins? Well, if you don't control powdery mildew, the fruit is not going to have as good color. You can see this one's not quite as dark orange, but much more importantly, the vines are going to die back early, and therefore the handle is going to turn brown and start to rot. And people like to, when they pick up their pumpkin using the handle, or technically the peduncle, and want to have a nice attractive handle. So that's a big reason to control powdery mildew with pumpkins. With other crops, it's even more important. With melons and winter squashes, you don't have as much sugar stored when the vines die down prematurely. Melons are not going to have flavor. Winter squashes aren't going to store as well. So a lot of reasons to control powdery mildew in all the cucurbit crops. So when I started, the first thing I wondered is, could poor control be because of fungicide resistance? I knew the pathogen had already developed resistance to benlate, which was the first targeted fungicide that had been developed. That's a FRAC1 fungicide. FRAC groups that stands for Fungicide Resistance Action Committee. Different chemistry goes into different chemical groups, and they're called FRAC1, etc., of different numbers, for those of you that aren't familiar with FRAC codes. So the product that growers were using at the time when I started that had stopped giving them good control was Balaton. That's in a different FRAC group, FRAC3. It's the only targeted fungicide they had to use at that point in time. And I was looking through some efficacy data that other university professors had published, and was seeing some early poor control in the late 1980s, towards the time that I had started here at Cornell. And then the other thing that really had me thinking fungicide resistance is, while I was at Penn State as grad student, I had volunteered to be the AV person so I could attend this big international fungicide resistance workshop that was conducted at Penn State. So fungicide resistance was definitely on my mind at that point in time. So we needed techniques. So first up was to develop techniques for looking at fungicide resistance. Powdery mildew, it's an obligate pathogen. So the only way we can maintain it is on leaf tissue. Develop this technique taking cotyledons, putting them on water auger. Sometimes they'll actually root there, and we can maintain the isolates right here directly on the cotyledons. Which is the first kind of interesting thing about powdery mildew, because if you look at cucurbits growing in the field, you will never see powdery mildew on cotyledons. You won't see powdery mildew until the plant is stressed, and that's typically by fruit production. So very interesting that in the lab we can work with powdery mildew on cotyledons. Technique we developed was this laboratory leaf disc assay. We basically take plants at the cotyledon stage, spray them with different fungicide solutions, then the next day cut out discs from those cotyledons, put them in these quadrant petri dishes on water auger, give them 7 to 14 days for the powdery mildew to grow, and then look at the severity on our control discs compared to the fungicide treated discs. So that's the whole technique. And it works pretty well because the cotyledon of cucurbits is a nice fat tissue, so it really survives quite well on these discs. So here's the situation when I started. Resistance had probably developed to Balaton, active ingredient tridemophan, in around 86, so about 2 years after the product was registered for use. That's pretty quick. And no, these dates are not accidentally reversed. Resistance had been detected in the pathogen 5 years before the product benlate actually made it to the market. And that was detected in Geneva. So I'm kind of was carrying on the tradition of Cornell being involved in looking at resistance in this pathogen. So we clearly didn't understand much about resistance back in 72 when benlate was registered. It was looked at, maybe this was just an oddity that we were picking up isolate. Well, the scientist who was working on it before me had picked up isolates that were resistant. Didn't know what it was going to do for control. Certainly an awful lot we didn't know back then about fungicide resistance. So one thing to notice about cucurbit powdery mildew is it's really happy on the underside of the leaves. You can see the colonies here are much larger than on the top side of the leaf. It's going to develop a lot better there. And this is where these new targeted fungicides were really giving us benefits in managing powdery mildew. They're able to move to the underside of the leaf and control the powdery mildew there. So if you were to spray a pumpkin crop or any cucurbit crop with a contact protectant kind of a fungicide like copper or chlorothalonil, you're going to get control on the top side of the leaf, but very poor control on the bottom side of the leaf. And that's because with these big leaves, the spray deposit lands on the top side. It's really hard to get it to move to the underside of the leaf. And these products can't move through the leaf. So if you don't control powdery mildew on the underside of the leaf, this is what the leaf is going to look like too soon. It's going to die. No powdery mildew on the top side, but plenty on the bottom, and it's causing the leaf to die prematurely. So we're quickly finding out with benlate and then Balaton and the targeted fungicides we've gotten since is they are so important for controlling powdery mildew on the lower leaf surface. They're single site mode of action. This allows them to be able to move through the leaf. If you had a copper or chlorothalonil moving into the leaf, it's going to damage the leaf tissue. But these targeted fungicides can move through the leaf translaminally or systemically and not damage the leaf because of their single site mode of action. Unfortunately, that single site mode of action means they're prone to resistance. So back when I started in 1988 into 1990s, I'm starting to really look at fungicide resistance. We had the foundation for general recommendations for managing fungicide resistance had come into play then, and they're really what we have today as our foundation for managing fungicide resistance. So first and foremost, having an integrated management program, so you're reducing your need for fungicides. The number of pathogens, you could use rotation. Doesn't work with cucurbit powdery mildew. It produces a wind dispersed spore, gets around very easily. So rotation is not just a play. When I started, we did not have much for resistant varieties. They've been developed a lot since. So not much else we could do for reducing our need for fungicides with cucurbit powdery mildew. Next general recommendation is to alternate amongst single site mode of action chemistry so that you've got different chemistry out there to kind of slow down that resistance development. Well, at that point in time when I started, all we had was Balaton and benlate, and we had resistance to both. So we didn't have a solution there. The next is to combine our at-risk fungicides with our broad spectrum fungicides, chlorothalonil, copper. So that's our general resistance management program. Out of luck back in 1990 for what we could do. So solution I was able to do for growers was to obtain what is called a Section 18 emergency registration, and I was able to get it for 2 fungicides that were then effective for powdery mildew. One was a new FRAC 3 fungicide, so that's in the same group as Balaton, but it is inherently more active, so it was effective against powdery mildew. And then a brand new chemistry that was just coming on the market, Quadris, which is a FRAC 11 fungicide. Since then, EPA has said they will no longer accept applications for Section 18s for 2 new fungicides. So that was a very unique time, but it did enable growers to control powdery mildew. A little bit about some of the other research that I routinely do. One is evaluating fungicides, and I have a tractor sprayer, as you can see here. I've had a couple over the years, so that I am spraying similarly to what a grower would be doing and having a big plot. Part of my goal is to see how well the at-risk fungicides are performing over time. So I will test them each year, just applying them every single week. Gives me a chance to see, as resistance is developing, is it affecting control or not? But it's also important just to be evaluating fungicides for growers. And that's because registration of pesticides, unlike when a new drug comes on the market for managing diseases in people, the efficacy of the product is not considered in registration. So it's important to be able to get this information for growers, and important for me to be able to come up with the best management programs for them. And I found sometimes a product we think is going to be really super effective is not. And actually, one of the most recent examples was a new FRAC 3 fungicide that was thought inherently to be a lot more active than the other ones that came on the market. And I found that it was not. And so it is for some of the other diseases it's been looked at, but just to show you, there's a lot of reasons to be doing fungicide evaluations. First and foremost, as I said, for seeing when resistance is impacting control. Another technique that I developed was doing the seedling bioassay, and this entails treating seedlings with different fungicide concentrations, put them out in a commercial crop or next to my research plots for a day, to have spores dispersed on them. And then we bring those seedlings back to the greenhouse. And about 10-14 days later, we've got enough powdery mildew on the leaves to rate them. So this gives us a quicker response, quicker data than the LabDis assay. But what it's doing is it's showing us what's happening in the population. So I can't see what's happening with an individual, which is the advantage of the other technique. So here we are with some results from quite a few years ago. And, another just really interesting thing about powdery mildew. Look at all that powdery mildew on that non-treated leaf. Those plants were out in the field for about 6 hours. And to me, it's just amazing how disease that leaf can get in just that short amount of time. But you can see here, just seeing kind of a range of different sensitivities, or just a degree of amount of resistance that's present. So at that point, picking up a lot of resistance to FRAC code 11, some of the DMI fungicides not doing as well. And at that point in time, we were not seeing resistance to boscalid or Quintec, just to show you this whole range. This was a trial I had done in Pennsylvania. This was kind of a year that it was kind of a giving back period, in that when I looked at this job as a grad student, I didn't know much about extension. And I went and talked to the vegetable extension pathologist at Penn State and kind of got a feel for what extension was about, and said, "Boy, I really like this." Well, just before 2008, he passed away due to cancer, and there was a void in their vegetable program. So I tried to do a lot of outreach activities with Pennsylvania extension specialists, kind of my give back to to Penn State for my grad school days. So let's jump ahead, fungicide resistance and kind of all the things I've seen over the years. So here we are of all the different chemistry, see all these different FRAC codes that have been registered over the years up until 2018. A lot of interesting stuff. So Quadris came on the market. I mentioned that one where I'd gotten a Section 18 for it. When it first hit the market, the company, Syngenta, thought, there's really low chance we're going to see a resistance problem with this chemistry. We don't need a resistance management program. They didn't have any restrictions on the label on use. And look at this. 3 years. We picked up resistance to powdery mildew in 3 years. And actually, it was the second pathogen. Resistance was picked up to the pathogen causing gummy stem blight in cucurbits before the powdery mildew pathogen, which was thought to have been the first. That was very interesting. It was also a particularly interesting year because I was on sabbatic leave in Australia and decided I'm going to do keep a little bit of my research program going. And one of them was maintaining the fungicide efficacy trial. And that's where we picked up resistance and control failure. We've had a lot of FRAC3 DMI fungicides come on the market. And we don't have, except for that first one, Balaton, we have not had control failures. Some of the best chemistry now, some of the most effective fungicides on the market now are in that group. So it appears the pathogen can't develop enough resistance to this. It's a multi-gene resistance as opposed to the single site mode of action resistance to the FRAC11 and the FRAC1 chemistry. So that stayed on the market. First SDHI fungicide that came on the market, we had resistance in 6 years. Quintec hit the market. Note when Quintec is here, first hit the market, we did have Pristine and we did have DMI fungicides we could alternate with. We didn't have resistance yet, still got resistance. So we've got alternation ability, but we're not completely preventing resistance from developing. Maybe we're slowing it down. Torino was a particularly interesting one for resistance to hit. We picked that one up in 5 years. That one, the company decided to have a very robust label for resistance management, only allowing 2 applications per crop. I would have thought that would have helped really slow down resistance. We have some other chemistry that's remained on the market and being effective. SDHI, Luna series, fluopyram is chemically a little bit different from the other SDHIs. And I'll talk a little bit more about that in a minute. So I'd mentioned that the broad spectrum products like chlorothalonil and copper are going to give you good control on the top side of the leaf. Here's a crop back when Quadris resistance was first picked up. You can see crop looks okay, a lot of green leaves. So growers were looking at this. This is great. We're getting decent control. So it's a fair amount of education I needed to be doing of the importance of you got to look at the underside of the leaf. So there's an underside of one of those leaves, white as snow. Bravo's working great on the top side of the leaf, no control on the underside where Quadris is acting all by itself. The SDHI fungicide. So I want to talk a little bit about how we've seen some cross resistance with some chemistry and not with others. So I've done some work where I've tested multiple SDHI fungicides. So this is with an isolate that is resistant to that first SDHI FRAC7 fungicide that came out, boscalid. So it's able to grow on boscalid discs with no problem at all. It's also able to grow these isolates on discs that have been treated with 2 other SDHI fungicides, but not with fluopyram. So fluopyram binds a little bit differently, so there isn't complete cross resistance. These are the kinds of things that are really important to know to be able to advise growers, what other fungicides can you use. In this case, none of the other SDHI fungicides except for fluopyram. Another thing I've been finding a lot of recently that is pretty alarming is isolates with multi-fungicide resistance. And we're detecting this through that leaf disc bioassay by testing isolates to multiple different chemistries. So I have frequently been finding isolates that are fully resistant to FRAC1, which isn't being used anymore, which is pretty darn interesting that resistance hangs around. So no fitness cost. And I think you can see from these discs that the pathogen is growing quite well on fungicide treated discs as well as on the control. I don't see fitness costs. So fully resistant to FRAC1, to the boscalid of FRAC7, which would also be some of those other cross-resistant FRAC 7 chemistry, FRAC 11, FRAC 13, and U6 fungicides. So that's a little unnerving when it comes to management, because using any one of these fungicides is going to be selecting isolates that are resistant to the others. I do find some isolates that are only resistant to a couple different fungicides, not all of them, but it means your alternating program is just not going to be as solid as we'd sure like it to be. The other thing I wanted to point out is by fully resistant, what I mean is in those fungicide bioassays, in the lab disc bioassay, I am using, and also in those field seedling assays, what we're applying in the lab is the full field dose. In other words, the concentration that is in a grower spray tank. So they are fully resistant. I'm often finding, particularly recently, that these isolates that are fully resistant also exhibit some reduced sensitivity to our newer FRAC 3 fungicides, the fluopyram, and also our FRAC 70 fungicides. So to give you an example, here is a sensitive isolates from some recent assays that we've done. So you can see it's growing fairly well on the control discs. Occasionally we do get an escape. I don't consider this to be tolerating that concentration. We require seeing growth on at least 3 discs to say that it is resistant. So you can see absolutely no growth on any of these other chemistries that were being tested. And here's a resistant isolate. So it's growing well on Torino, FRAC U6, FRAC 13 Quintec, Andorra FRAC 7. It's also growing on Aluna Privilege and Raleigh at the same concentration that was tested with a sensitive isolate. Those are partially resistant, not fully resistant, in that those are not at field capacity, field dose rate. So it's a slightly higher, a slightly higher rate than what the sensitive isolate can tolerate, but not enough that we're seeing an impact on control. So there is the question of whether we're going to continually see shift with some of this chemistry or not. Always more to do. The other thing I've done is to collect isolates from growers' fields to try and get a feel for presence of resistance in their field. And this is more recent work trying to see, do I see shifts, potential for shifts because of what they've used? And what I want to point out here, a couple growers where they've used Quintec twice in rotation, and they've got 67 to 100% of the isolates from those fields that we tested were resistant to Quintec. Whereas the field where just protectants were used or Quintec once, we're picking up no resistance to Quintec. And then the other thing you will see is some high resistance percentages to the FRAC 7 fungicide Endura, although no FRAC 7 chemistry has been used here, and that's because of that multi-fungicide resistance. So these isolates that are resistant to Quintec are also resistant to Endura. So back to that comment I'd made earlier about the fact that we got a lot of powdery mildew on the underside of the leaf, not getting control with protectant fungicides there. Next question was, is there any way we can get our protectant fungicides to the underside of the leaf better so that we can get better control? They can have more of a role in resistance management. So I invested in an air assist sprayer. I did some work with the technology person who was at Cornell in looking at different nozzle types, air induction, twin jets on a regular spray broom, plus this air assist sprayer. We just tried all kinds of things to improve control on the underside of the leaves. And I looked at it in 2 ways. I had plots that were treated with just Bravo. Can I get better control on the underside of the leaves? And we also did the very simple procedure of putting water sensitive paper on, placing it on the underside of the leaf or on the top side of the leaf to see what we're getting for coverage. And you can clearly see we're not getting much spray on the underside of the leaves. It didn't matter with that air assist sprayer. We were not driving more spray material to the underside of those big pumpkin leaves, unfortunately, and it matched the control. More recently, biopesticides have come on the market, and I've gotten involved in testing them. There are quite a few that are labeled for cucurbit mildews, both powdery mildew and downy mildew, some of them. A number of them have a microbial active ingredient. There's a bunch that have a botanical active ingredient and some that are just natural products. As I mentioned earlier, EPA does not require efficacy data for registering any of these, thus the value of testing them and seeing how well they work. And bottom line of what I found with powdery mildew is they have contact activity. I'm not seeing control on the underside of the leaves. A number of these products, in particular Lifeguard and a number of others that have bacillus, they are being promoted as inducing resistance. If they were inducing resistance in pumpkins or any other cucurbit, I think I'd be seeing control on the underside of the leaf, which I'm not. I think an interesting next step could be for somebody who's got the capability of looking at the plants and seeing, is resistance actually being induced? I think that would be a very valuable next step with some of these products. I see them from my work as being really helpful for organic growers. They're giving them a better solution than using copper. I'm still fighting to try to find how they really fit well for a conventional grower. So I also look at other ways to manage powdery mildew. There are resistant varieties, more so have come on the market in more recent years than when I first started out. Here's an example of a variety trial for varieties that were resistant to powdery mildew in melons. What I found with melons is there is really strong resistance. It's a race-specific resistance. If you've got resistance in the melon to race 1 and 2, which have been the dominant races that I've found when I've done screening of races, we're not seeing a big race war, thank goodness, with powdery mildew, not seeing some huge changes over time.
I think my dad did it too.
Is there a question? Okay, I thought I saw heard some other talking. I see really good control with the resistant melon varieties. Growers need to know what kind of control level they're getting with their resistant varieties to know how it fits in a program. I found with squashes and pumpkins, I get very little control. So a grower really needs to have an integrated management program where they're using fungicides as well. Just an example to show you susceptible variety and the resistant varieties, kind of, you can see sometimes a range in how well they control powdery mildew. But it's important for growers to know the quality of the fruit because that's what they are selling. The trials that I've done, I interact with the extension specialist, cooperative extension specialist here in the county. She helps me with evaluating the fruit quality, so it's a good interactive program.
1 second.
Yes.
Somebody has unmuted the microphone. Please mute your microphones. Okay, Meg, please go on. Yeah, sorry.
Okay, no problem. Growers need to know the quality of the fruit. So that's the additional kind of things we do. And I'll kind of point out with this one, because this is just a fabulous result. Trifecta was rated the highest for overall fruit quality in our evaluation in 21 and also the past year. That comes from the Cornell Plant Breeding Program from Michael Mazurek. So proud to see that result. Some other research activities I've had with powdery mildew over the years. At one point in time, I had someone working with me in my program who graduated from Cornell, focused on mycology. And she helped correct the name of the pathogen to Podosphaera xanthii. We also noticed that the pathogen's sexual stage was present often in the fall. Chasmothecia, you can see these little tiny black fruiting bodies on the bottom side of the leaf. And we discovered that we could get them to appear on leaves in the lab. And actually it's very interesting. Once again, another interesting thing with powdery mildew, while we don't see this until the fall, often after we started getting cool temperatures at night, you put two isolates together that are of opposite mating type, and they'll produce those chasmothecia pretty much as soon as they've met each other. Able to document as we proceeded with this work that they're heterothallic, looked at occurrence of both mating types and found that they commonly do occur together in a field. At that point in time, I had gotten some isolates from other areas of the US and found the same thing. my mycology expert partner at that point in time was interested in looking at whether the ascospores survived over winter. She was able to document that indeed in leaf tissue they do survive. There are some viable ascospores in the spring. They kind of do decline a little bit over the winter. But it's kind of important to think about the whole overall biology. And I do wonder about the role in the life cycle, because a grower is going to plow down their field in the fall typically. That's putting the leaf tissue into the ground, coming back with a cover crop. And most growers are not putting cucurbits back in the same field the next year. Another really interesting thing about powdery mildew is its host range. It also goes to verbena. They're not related. I find this just fascinating. I took powdery mildew off of verbena and moved it onto pumpkins, and it grew just fine, just happy, verifying that indeed verbena is an alternate host. That's important because if you're growing cucurbit seedlings for sale to home gardeners in a greenhouse where you're also growing verbena, you could end up with powdery mildew on those plants you want to sell to gardeners. And it's also important because sometimes there are different fungicides registered on ornamentals, and might be getting some early selection for resistance. Well, I've had a lot of work. Every year I've had experiments on cucurbit powdery mildew, and there's still plenty of work left to do, although this was my last field season this past year. I've also had time to look at other topics. One I got into right away when I came to Long Island was looking at the impact of ambient ozone on plant productivity. Here out on Long Island, we do see high ozone every summer, and that's because of the car traffic that we have. And it was this work I did was part of a regional team of other scientists. Here is a line. We worked with two lines of beans. This is one that is not sensitive to ozone, and we had a line that is very sensitive to ozone, just to see how much symptoms we get, how much is impact. And the impact that I saw on yield was up to and over 50% reduction in bean yield, which is pretty phenomenal. I've also worked on cucurbit downy mildew after its reemergence in 2004. We believe a new strain of the pathogen that came in at that point in time, which was able to overcome the resistance in cucumbers, and also had resistance to some fungicides. So all of a sudden, the major ways that downy mildew was being managed, which was resistant cucumbers, which was the main crop that was being hit, and also fungicides, we've lost those controls, and downy mildew has been a major issue ever since, and in all of the cucurbit crops, not just cucumbers. So some of the work I've done is looking at efficacy of fungicides, looking at biopesticides, evaluating resistant varieties, so growers know how well cucumber, and there's also a few cantaloupe varieties, are able to resist downy mildew. I've looked at integrated programs, so combining conventional fungicides on a resistant variety, or combining biopesticides with conventional fungicides in looking at kind of an integrated program for growers who might want to try to reduce their use of conventional fungicides. I've been involved with a national team monitoring the occurrence of cucurbit downy mildew. This is a fascinating pathogen in that we know it can survive over winter in South Florida with the winter production of cucurbits there. It is not known to be surviving by oospores elsewhere, and we can monitor and track its movement up the East Coast every year. And to me, that's just phenomenal, a pathogen able to move that far every year, often making it all the way into Canada. Thank God COVID didn't move that easily. And I've also looked at fungicide resistance. So you can see this is the seedling bioassay is the way I've looked at fungicide resistance in cucurbit downy mildew out in a commercial field. So you can see the impact of downy mildew on cucumber, and downy mildew on cantaloupe, or the two leaves to the side. Basil downy mildew. New disease first detected in the United States in the fall of 2007. I've looked at efficacy of fungicides, biopesticide, resistant varieties, which have had their place and done a really good job. Some of them. Here is a variety that has no resistance. susceptible variety. Here is one of the resistant varieties I've looked at that has a very high level of control, but it's a single gene. And with that one, we are starting to see breakdown of its resistance, starting to see a new race of the pathogens starting to appear. So kind of wondering if we're going to be on a treadmill of trying to keep up with having resistant varieties that can stand up to basil downy mildew. There are some other varieties that have a multi-genetic resistance. So we do see downy mildew on them all the time, not anywhere near as much as a susceptible variety. And that's a more durable resistance. So we'll see what happens over time. Challenge with a disease that's affecting an herb or a leafy vegetable is this fresh market is it doesn't matter if you got a lot of the pathogen on it or just a little bit, those leaves are unmarketable. So you've really got to have a high level of control. And I think that's where very difficult to get control with biopesticides. I'm more looking at biopesticides applied to a resistant variety to try and see some control. The other thing I've been involved with since it started having this disease is monitoring its occurrence in the US. This is my most recent monitoring program that I've got is with the Ag Pest Monitor group. And that's been interesting for just learning about its occurrence, gaining more knowledge about the disease. One interesting case in particular, quite a number of years back, kind of early in its occurrence in the US, we're kind of wondering where this disease had come from. I guess I should back up and say, prior to detecting it in the US, it had been detected in Europe, starting in around 2001. And before that, it was detected in Uganda in 1930, 33, 34. So it's kind of this mystery disease. What had it been doing all those years? You know, why hadn't it gotten anywhere else? One of my reporters was from South Africa, and it was after the International Plant Congress, and I had met some people from South Africa, so I contacted them, tried to get a hold of somebody there just to learn a little bit more about basil downy mildew occurrence in South Africa, and learned from their extension person that they'd worked with a grower in the late 1990s who was marketing basil to Europe and having issues with downy mildew. So I suspect it went from South Africa to Europe and took off in Europe. It's a seed-borne pathogen, and that's how it got to the US. There's also been a number of new diseases that I haven't done research on, but have done extension on. One of them is Stemphillium. I've had actually a couple Stemphilliums, but Stemphillium leaf spot in tomato, showing here in contrast with septoria leaf spot symptoms, a more common disease for us. Stemphillium is a disease that occurs primarily in the southeast of the United States. It likes it warm and humid. We started seeing it very recently in high tunnel production tomatoes, which is a new way that we're growing tomatoes in the Northeast in recent years. Also seeing a little bit of it starting to appear in the field. So this may be a disease that with climate change, we're going to start seeing more commonly. Some of the other new diseases that I've seen in recent years have come in on seed, particularly organic seed, and that's been a big issue. Late blight. I started working on that after its reemergence in 2009. Interestingly, in recent years, it's kind of disappeared again. So that was kind of the end of working on it. You can kind of see here what happens with a susceptible variety. If you don't apply fungicides, a resistant variety, we can get some good control. I think interesting for me with this one is just watching it spread. In particular, in 2009, that was an outbreak that started because a company that sells plants to home gardeners had an outbreak in their plants, and they were selling those plants. That was interesting, but 2011 was actually the most interesting year on Long Island in that we had one company in New England produced plants for one garden center on Long Island, and they had started tomatoes and also potato plants for home gardeners. Their potato plants had late blight. It spread to the tomatoes. They culled out all the tomatoes that had symptoms, sold the remaining plants to a garden center on Long Island, and that one source led to a huge outbreak on Long Island. It was actually even worse in 2011 for us than 2009, and it was just interesting to watch the spread of this pathogen and disease on Long Island that year. So a lot of interesting things over the years. The other one I've worked on is Phytophthora blight. Really started taking off in the 1990s. You can see what it does to pumpkins. That's been really where it's hit the hardest. Also it causes a crown rot on pumpkins, blight, hits other crops as well. I've looked at reduced tillage for managing Phytophthora blight, and I've looked at mustard biofumigation, as well as fungicides, biofungicides. Another new interesting that happened during my career is seeing Phytophthora jump to a new host, snap bean, not related to cucurbits or peppers or eggplants, which are the other hosts. Here's symptoms on snap beans. I've also done research on foliar diseases of tomatoes, fungicides, biopesticides, resistant varieties. Fair amount of work in recent years since Tom Zitter, who was in Ithaca, had retired, working with the Cornell tomato breeder in evaluating her experimentals. So hopefully I've given you a good feel for how interesting cucurbit powdery mildew has been and how well it's been able to fill my career. It's also been really rewarding. It's been rewarding just advancing science, having colleagues thank me for the work I've done. But as I look back over my career, to me, the biggest reward, what's given me the greatest satisfaction, is when other extension faculty, cooperative extension specialists, but particularly growers, thank me when I answer questions or give a presentation and they've liked the work. And when I see the amount of hits to websites that I've got with extension materials, to me, that's been the most rewarding. So I guess for me, an extension position was really valuable, and I'm really thankful for the time I've had in this position. And I need to acknowledge that I've had a lot of people help me out over the years. I haven't had grad students because I'm so far from campus. I've had several technicians over the years. Currently, Colin is the technician who's working with me. He's my eighth, last, best in a lot of regards technician. One thing, shout out to Colin. I know you're on the line, and I hope you're not blushing there, but to me, what's been really interesting is his background is not in plant pathology. He has graphic arts as a background that makes him very detail oriented. He's been very quick to learn. So it's kind of a message that you can excel in a position even though that position is not your background. So I'm very grateful to Colin and my previous technicians for their help. And I've also had a lot of summer students join me, usually college students. This is Claire from this past year. She is a recent Cornell grad, which is why I wanted to include a picture with her. And I think we got a few minutes for questions, so I will stop sharing.
So Meg, thank you very much. It was wonderful and very nice to hear from you. Very good snapshot of a lot of work you have done. So now we are open to questions, and there are some questions. I think I saw a question from Juan in the chat, but also anybody in Geneva or on Zoom, if there are questions, please unmute and go ahead and ask.
So I can start with the first question. So you want me to go ahead and read it? I guess I could read it. So given the evidence of fast multi-fungicide resistance stably stacking in some of these isolates, plus the non-renewable character of most plant resistance genes, mechanisms, it kind of feels like the efficacy of management strategy is slowly eroding. Yes, I do agree. Where do you believe we should be focusing now before we get to the point where we were before the 50s? We do get good control now with the fungicides we've got. It is a question, will we see resistance to all of those? We're seeing new chemistry coming out, so I'm hopeful. The big is having resistant varieties, and that's where I'm hoping that gene editing, precision breeding is going to become acceptable, and we will see advances in better resistance hitting the market. I think that's going to be a real panacea, because you can take a variety that has all the characteristics a grower wants and edit it, instead of crossing and then trying to cross out all the bad characteristics to get back to get an acceptable variety. And I've seen that in evaluating the material from plant breeders, particularly when I'm working with plant breeders and early material. Often there are some issues with some undesirable traits. Basil, for instance, with downy mildew, the parent was a spice type that had a licorice flavor. Took time to breed out that licorice flavor. Hopefully that answered your question. Speak up if it didn't.
Yeah, I think Chris Smart has a question, and very.
Hey, Meg.
Hey, Chris.
Good to see you. Great presentation. One of the things I guess I wanted to highlight to the group, and then a question for Meg. The fact that Meg is on Long Island, it's a huge bonus for those of us who are in Geneva and Ithaca, because the agriculture in Long Island is so incredibly unique. The soils are unique, the growers are unique, the amount of traffic is, everything about it is different, which has been great for us. It's great for us to have those folks there. And Meg has done an amazing job. I guess my question, Meg, is how do you see ag in Long Island? I guess I see Suffolk County as continuing to be a powerhouse in agriculture, but I guess I'd appreciate your opinion, and how has disease control changed because you're on Long Island and all of the requirements of Long Island?
Well, that's a big question. I think we'll always have ag here.
I agree.
First of all, the first land preservation programs started here on Long Island. So we have a lot of land that is preserved from development. We have a lot of people who want to come out and come out not only to buy produce from the farms, also for the ag entertainment. We now have a lot of wineries, distilleries, breweries, people coming out for that. Some of the biggest greenhouse operations are here on Long Island. We've got nursery, we've got sod, a lot of diversity. We're, I think, past capacity for people, but there certainly isn't more room for people. So we need to keep the land and ag from that perspective. But seeing people contributing to land preservation, there is a farm in my town where people contributed funding to preserve some of the acreage and keep that land. It was a farm that had been split between the 3 children. So 2 of the children wanted to develop their chunks, and we're looking to sell it for, I think it was $500,000 per acre. So you're talking a lot of money that people put into preserving that land so that it could be kept in the farm, some of the land. So we see those kind of things happening. We also have a lot of aquaculture. We still have a duck farm out here. My daughter, who's associate director with Long Island Farm Bureau, is continually reminding me of all the other agriculture that is here on Long Island. Yeah, there's a lot to do. The agri-entertainment in the fall, I don't leave my house on an October weekend. You're not going anywhere unless you're going to drive at like 6 a.m.
Thanks. Is there any other question?
I guess the other thing I'd add about Long Island is, given how different it is, I think that also adds to the addition of conducting research in New York, including Long Island. The fact that our soils are sandy, our climate is so much more temperate. We're an entirely different planting zone than upstate. And we're going to see impacts of climate change more so.
Hi, Meg, I have a question here in Geneva. Have you looked at the impact of foliar leaf surface coverage on fruit quality in terms of contact protectant fungicides and systemics, like if the upper surface is completely covered or completely controlled, the lower surface is covered versus coverage on both surfaces, do you see a substantial difference in reduction in fruit quality?
I've looked at it with cucurbits, and absolutely we do. We see, so the major one I have continued to look at is pumpkins, and we don't see fruit with as good quality. Our primary measurement is how the handles go down, so what percentage of the fruit remain with a good solid handle into early mid-September. And definitely the leaves die back and quality slips. The other thing that can happen if you don't have good a canopy cover, when we get into the fall, we have more dew periods, and you can get dew lasting on the fruit, and then you get an intense sunlight day, nice bright clear fall day, and you can end up with sun scald. So I've seen a number of fruits lost to sun scald because of not having a good canopy cover into fall.
So Meg, the question is, when you said that all the basil heat with powdery mildew could not be sold even if it's a small spot, is that because of ethics for spread, legal reasons, or customer preference for perfect looking products?
That's going to be a combination. That's downy mildew. First of all, in case I was confusing about that. Part of it is absolutely, I mean, do you buy leaves that have spots in the grocery store? You're probably going to avoid those. The other issue with marketing, you'll often see basil is marketed either in a bag or in a clamshell. That's a nice moist environment. A little bit of downy mildew is going to become a lot of downy mildew very quickly. So that's another issue.
Okay, so with that, I think first the announcement that anybody who wants to talk with Meg, just stay online and continue talking with Meg. But we should really applaud Meg for this amazing and beautiful presentation, and her career is really amazing. So yeah.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.